Hello students, this is Dr. Tikia Arashiro. In this podcast, I'm going to talk about the regulation and transcription of genes involved in porin synthesis in response to osmotic pressure. So let's get started. Porin regulation is a two-component regulatory system widely distributed in bacteria and particularly well characterized in Escherichia coli. Its function is in osmoregulation, which responds to changes in environmental osmolarity by regulating the expression of the outer membrane porins OMP-F and OMP-C. ENVZ is the sensor kinase, which is the osmosensory domain, and the OMP-R is its corresponding response regulator protein. So let's review porins. Porins are channels that allow the diffusion of small hydrophilic molecules. They cross the cellular membrane and act as a pore through which molecules can diffuse. Unlike other membrane transfer proteins, porins are large enough to allow passive diffusion and act as channels that are specific to different types of molecules. They are present in the outer membrane of gram-negative bacteria and some gram-positive microbacteria, the outer membrane of mitochondria, and the outer chloroplast membrane. Porins were first discovered in gram-negative bacteria, but they have also been found in gram-positive bacteria. They exhibit similar transport functions but have more limited variety of porins compared to the distribution found in gram-negative bacteria. Gram-positive bacteria lack outer membranes, so these porin channels are instead bound to specific lipids within the cell walls. Archaea also contain ion channels that have originated from general porins. The channels are found in the cell envelope and help facilitate solute transfer. They have similar characteristics as bacterial and mitochondrial porins, indicating physiological overlaps over all three domains of life. This podcast will focus on outer membrane proteins in gram-negative bacteria whose levels are regulated due to the changes in osmolarity of the environment. These outer membrane proteins are OMP-C and OMP-F porins. Now, OMP stands for outer membrane protein. So OMP-C and OMP-F proteins are porins that allow metabolites to cross the outer membrane of gram-negative bacteria. OMP-F forms a large pore than OMP-C. Now, the osmolarity or osmotic pressure in the environment controls the relative levels of OMP-C and OMP-F in outer membrane. It is important to note that OMP-F and OMP-C proteins are expressed at all times at constant levels. However, the individual porins OMP-F and OMP-C are present in various relative abundances based on medium osmolarity. So when the osmotic pressure is low, the synthesis of OMP-F, which is a larger pore, increases. And on the other hand, when the osmotic pressure is high, the OMP-C protein, which has a smaller pore, is made in larger amounts. The OMP-C proteins are present all the time, but present in large amount when osmotic pressure is high. Gram-negative bacteria respond to changes in osmotic pressure by regulating the synthesis of these two porins, OMP-C and OMP-F. And to respond to changes in osmotic pressure, they use the porin regulation system, which is part of a two-component regulatory system. So the ENVC protein is a sensor transmitter that spans the plasma membrane and has historically been divided into two domains, the sensory and the transmitter domain. The protein is composed of a short N-terminal tail in the periplasm and a cytoplasmic domain containing the autophosphorylated histidine residue. So the ENVC protein is a sensor kinase that serves as an osmotic sensor. It detects changes in osmotic pressure. When a shift in osmotic pressure occurs, it autophosphorylates and transfers its phosphate group to the response regulator OMP-R. The OMPR protein is the response regulator. It is located in the cytoplasm. It is a DNA binding protein that regulates expression of OMPC and OMPF genes. Variations in osmolarity trigger conformational changes in ENVC protein, which affects phosphoryl transfer to OMPR. Upon phosphorylation, OMPR becomes an active dimer that exhibits enhanced DNA binding ability specific for both the OMPC and OMPF genes. The ENVC and OMPR interact to bring about the differential regulation of two genes, OMPF and OMPC, which are responsible for the production of outer membranes porin OMPF and OMPC. Under conditions of low osmotic pressure or low osmolarity, ENVC protein exhibits comparatively low kinase activity towards OMPR. Hence, the amount of the phosphorylated form of OMPR in cells is relatively small. In this particular situation, the phosphorylated OMPR complex binds cooperatively to the activator sites of OMP gene and upregulates its transcription. These sites have high relative affinity for OMPRP, or phosphorylated OMPR complex, as compared to the C1, C2, and C3 activator sites of the OMPC. Therefore, the transcription levels of OMPC is less as compared to the OMPF under low osmolarity condition. This phenomenon has been attributed to the fact that OMPF has a larger pore diameter than OMPC, which results in tenfold faster diffusion rate that provides scavenged scarce nutrients. So upregulation of transcription OMPF leads to the synthesis of more OMPF protein. Conversely, when osmotic pressure or osmolarity is high, the ENVC protein actively undergoes autophosphorylation and then efficiently transfers its phosphoryl group to the N-terminal receiver domain of the OMPR protein through NVZ OMPR complex formation. As the number of phosphorylated OMPR protein molecules increases, two events occur. The response regulator OMPR binds to the high affinity binding sites upstream of the OMPR promoter, but also to the one low affinity binding site. Binding to this low affinity site results in repression of OMPF gene. So at high osmolarity, increases in phosphorylated OMPR represses transcription of OMPF gene and activates transcription of OMPC instead. So how is the expression of OMPF and OMPC regulated at high osmotic pressure? High osmotic pressure activates ENVC so that it undergoes what you call autophosphorylation. Then it phosphorylates the response regulator OMPR. So the phosphorylated OMPR accumulates in the cell at high level. The high level of phosphorylated OMPR not only stimulates OMPC gene expression, but it also represses OMPF gene expression. As the number of phosphorylated OMPR protein molecules increases, phosphorylated OMPRs binds to the operator downstream the OMPF gene promoter, blocking transcription of OMPF gene. Further, the phosphorylated OMPR binds to the low affinity activator sites upstream of the OMPC promoters, and as a result, OMPC gene expression is stimulated and more OMPC porin protein is expressed on the outer membrane of the cell. Also, the transcription of MYC-F antisense messenger RNA is initiated at high osmolarity. MYC-F antisense RNA binds to complementary sequence of OMPF messenger RNA to complementary sequence of OMPF messenger RNA to block its translation. These two mechanisms ensure that there is relatively higher expression of OMPC as compared to OMPF under the conditions of high osmolarity in the media. So high levels of phosphorylated OMPR repress OMPF gene but stimulate OMPC gene expression. At high osmotic pressure, or when the concentration of solutes in the medium is high, the OMPC gene expression is stimulated, and so more OMPC proteins are expressed in the outer membrane of the cell. The high number of OMPC proteins on the outer membrane allow the entry of more solutes into the cell, thereby maintaining equilibrium and preventing the cell from shrinking due to water loss. Recall that when solute concentration outside the cell is high, it builds osmotic pressure in the cell wall, so smaller polar molecules in the cell, like water, will leave the cell and move towards higher concentration of solutes, and so the cell shrinks. So the higher concentrations of OMPC prevents the cell from shrinking by taking in more solutes to maintain equilibrium. So what's up with MICF? What is the role of MICF in porine regulation? MICF is an antisense RNA that inhibits the translation of OMPF gene. It is about 93 nucleotides. Now, antisense RNAs in general are regulatory RNA molecules that regulates gene expression at post-transcriptional level. They are single-stranded RNAs, and their RNA sequence is complementary to the RNA sequence of the RBS, or ribosomal binding site, on the messenger RNA. Now, antisense RNA regulate various aspects of cell physiology by binding to other messenger RNAs and stop translation, and they exert their effects by base pairing with messenger RNA to stop or block translation. So how does MICF function in porine regulation? MICF is an additional mode of inhibition of OMPF messenger RNA translation, particularly at levels of osmolarity that are not very high. It binds to the OMPF messenger RNA, blocking its translation. This mechanism ensures that there is relatively higher expression of OMPC as compared to OMPF proteins under the conditions of high osmolarity in the medium. So how is the expression of OMPF and OMPC genes regulated at low osmotic pressure? At low osmolarity, the ENVC protein exhibits comparatively low kinase activity towards OMPR, and so the amount of the phosphorylated form of OMPR in the cells is relatively low. The low levels of phosphorylated OMPR in the cell prevents the OMPR from binding to the operator site upstream of the OMPF gene. This therefore stimulates the transcription of OMPF gene and the ultimate translation of the OMPF messenger RNA. And when the osmotic pressure is increased, the number of phosphorylated OMPR also increases, and this increase in phosphorylated OMPR represses the transcription of OMPF genes while stimulating OMPC gene expression. So upregulation of transcription of OMPF gene leads to the synthesis of more OMPF protein. So when the osmolarity is low, the concentration of solutes in the medium is low, and this stimulates the expression of the OMPF genes, and so more OMPF porins are expressed in the outer membrane of the cell. The higher number of OMPF proteins in the outer membrane allows the solutes in the cytosol to cross the outer membrane into the external environment to equilibrate the osmotic pressure and to prevent the cell from lysing. So when solute concentration outside the cell is lower than the internal solute concentration, water will enter the cell, causing the cells to swell and burst. The intent of osmotic regulation is to equalize the solute concentrations between the cytosol and the external environment to maintain equilibrium so that the cells do not lyse or shrink. So that's the end of the third podcast on responses to environmental cues. Stay tuned for the fourth podcast.